My name is Keith Mayer. I'm the Associate Director of Product Development at EpiCypher, and I'm excited to talk to you about a project we've been working on with DNAnexus for a number of years. So at EpiCypher, we like to think about epigenetics largely. And what is epigenetics? If you think about a zygote, it's a single cell that has one genome. And as that single cell turns into a whole person, you develop multiple cell types, right? And so the question that always, beleaguers people is, what is the difference between a heart cell, a skin cell, and a lung cell? It's really the epigenome. And what is that? It's the regulation of gene expression across those different cell types to create a symphony of different balances amongst the 20,000 different protein coding genes in the human body or any type of organism. And so largely the way I like to think about this is if you care about cells and why they might be different than other cells, then you should probably care about epigenetics. And because of that, it makes epigenetics relatable to a broad spectrum of research. You can care, we've found customer bases in developmental biology, oncology, immunology, but really the list goes on and on beyond here in terms of researchers who care about cells and should probably be caring about the epigenome. And when I'm talking about epigenetics, I'm really talking about the molecular biology machinery that is regulating that gene expression. And so here we're talking about DNA methylation on the DNA itself, histone post-translational modifications on the nucleosomes that the DNA wraps around, and chromatin proteins that can recruit down onto the chromosome and perform functions. And so all of these chromatin regulators exist in concert on the chromosome. And when you really boil it down, what it comes down to is the opening and closing or the winding and unwinding of DNA to regulate whether or not genes can be accessed and expressed. And when people want to study that open or closed chromatin, there are a variety of different ways to go about that. First top level is a binary view, wherein if you just want to look at accessible chromatin or inaccessible chromatin, you can use ATAC-seq or whole genome bisulfite or enzymatic methyl sequencing to get a view of the open or closedness of the chromatin. But if you really want to dive in and look at the epigenetic machines that are doing all the work, then you need to start to compartmentalize what they're doing. And historically, that's been done with ChIP-seq. However, ChIP-seq has its disadvantages. It takes a lot of cells to perform, which means it has a high cost, particularly in regards to its sequencing depth, largely because in ChIP-seq, you are cross-linking and lysing your cells, then you're fragmenting the chromatin and doing an immunoprecipitation, and that all comes with a really high signal to noise. It's also low throughput because you're constantly having to re-optimize your sonication to make sure you're getting the right fragment lengths. And it's technically challenging. It requires a really skilled set of hands in the lab in order to be successful in those assays. So at EpiCypher, really over the last 5 years, we've been really excited about two sister technologies, CUT&RUN and CUT&Tag. For brevity today, I'm only going to talk about CUT&RUN, but largely everything I say also applies to CUT&Tag. And in CUT&RUN, the main difference over ChIP-seq is that instead of lysing those cells, you're adhering them to a magnetic bead and permeabilizing the plasma membrane so that you can then float in the molecular biology reagents and do the enrichment of your DNA in situ against the chromatin as it exists in its native state. That chromatin that you enrich for then makes its way out of the cell. We can do standard library prep and sequencing. And then that's how you can really profile where these molecular machines are sitting on the chromosome in the relation to the entire genome. So CUT&RUN has some advantages that we really like over ChIP-seq, really by virtue of the fact that the cell stays intact and you're only liberating the DNA of interest. It has a really high signal to noise. And that value percolates down to the ability to use many fewer cells. So CUT&RUN is really able to profile precious samples that could never be before used in ChIP-seq. By virtue of that, it also allows you to reduce significantly lower, which dramatically reduces the cost of the assay overall. And because the cells end up on a magnetic bead, we're able to miniaturize the assay, and that allowed us to really increase the bandwidth that a single scientist could do in terms of using this technology at the bench. Also, EpiCypher at its core is a nucleosomes company historically, and we've leveraged that expertise to develop spike-in controls because one of the major linchpins for any assay that relies on an antibody is the antibody reproducibility crisis. And so we have designer recombinant nucleosomes that are DNA barcoded, bearing varieties of post-translational modifications on them so that you can, in the assay, monitor the specificity of your antibody to the target that it's promised to. And this is important because EpiCypher has found over the years that if you go randomly buy any commercial antibody to a histone post-translational modification, you have about a 70% chance of finding one that doesn't actually specifically bind the target of your interest. So over the last 5 years, EpiCypher has been working in this space to really bring a suite of technologies and products that can help researchers start to access epigenomics. And so now, where ChIP-seq was relegated mainly to expert epigenetics labs, we like to view ourselves as democratizers of epigenomics so that really anyone who cares about the epigenome of their cells can access these technologies and be successful at the bench. However, with increased throughput on the wet lab side, we've started to encounter a problem. And that problem is that with reduced technical barriers, meaning more researchers can access the technology, increased affordability, which means those researchers can afford to do larger experiments, and the fact that it's been miniaturized, which enables researchers at the bench to perform these assays with multi-channel pipettes or even deploy the assays on robots to really get these epigenetics going at scale, we're encountering a bioinformatics bottleneck. In fact, at EpiCypher alone, I just dug up the fact that over the last 7 years that we've been playing with these technologies, just internally for our research teams alone, we've generated over 30,000 reactions of CUT&RUN and CUT&Tag to date. And all of this data needs to get analyzed, which is causing a big bottleneck for many of our researchers. So today, we're really excited to announce that we've now closed the loop on the CUT&RUN and CUT&Tag pipeline by releasing the upcoming release of CUTANA Cloud, a bioinformatics platform that's going to really help people speed along their research. And so before I talk about what CUTANA Cloud does, I want to talk about the bioinformatics that really underlies this assay. And it's mainly broken up into 3 key parts. There's primary, secondary, and tertiary analysis. Primary analysis is usually taken care of by the sequencer. This is the conversion of images of the flow cells into text files that have base calls in them, or FASTQ files. Secondary analysis is the process of reference genome alignment, read filtering, and peak calling. And that really gives you your key output files of BAMs, BIGWICs, and BED files, which are the files that make their way into tertiary analysis, where you're doing the biological interpretation of that data to really gain insights into what happened in your experiment. And so just to walk through those 3 steps in a little more detail, for primary analysis, if you have your CUT&RUN sequencing library ready to go, you have your insert DNA, which is the CUT&RUN output, already gone through library prep so that it's prepped and ready to go on a sequencer. The sequencer is going to do paired end reading. EpiCypher strongly recommends paired end reading for CUT&RUN and CUT&Tag because it really helps you downstream. And once you have those reads off your DNA, they're assembled into a FASTQ file. FASTQ files contain anywhere from 5 to 10 million paired reads. So in these documents, each row of the read 1 and read 2 files are associated with each other so that we know which ones came off of the same cluster off the flow cell. Now the most computationally heavy step is you take these FASTQ files, and we also want to, so then we move into secondary analysis, where we want to know, okay, we got all these reads off the sequencer, where did they actually come from in relation to the genome of the organism that we did the experiment on? So this is the most computationally heavy step, where you take these files and you put them through a high-performance computer, and it's going to take each read pair and figure out where along the reference genome those sequences came from. And so now that you actually have one-dimensional spatial information about where these reads sit on the genome, with each of those paired read files, you can then infer the middle portion of those reads to recapitulate the full length of the input DNA that you got out of your experiment. And once you have your reads aligned to the reference genome, this is how you get your BAM file. The second step in secondary analysis is to filter. So we're going to remove duplicates that may have arose from PCR. We also want to get rid of reads that align to more than one spot in the genome so that we're only dealing with unique reads. And last, we want to remove reads that localize to regions of the genome that are known to cause false positive in these types of mapping assays. And once we get rid of those reads, then we're left with our unique BAM file. Finally, we want to reduce the complexity of this data a little bit and stop working with A's, T's, C's, and G's directly. And the 2 ways of doing that is, 1, to perform peak calling, wherein we use a program to computationally determine where the signal rises above the background. But as with many things in science, there is also art to it. And so there's a little bit of thought that has to go into deciding which peak color you use, because based on the peak color and the settings that you use, you can actually generate different profiles of peaks out of your data. And so it's really important to sit and think about, what is the expected structure of my data? What am I looking to, what types of peaks am I expecting there to be? And then on the back end, make sure that those peaks actually match up qualitatively with the data that you have. Because as with many things here, just getting more peaks out of your sequencing does not necessarily mean that you had better data. So that gives you your bed file. And then lastly, we can also smooth out those tracks to an area under the curve. And then that generates bigwig files that can be used for generation of genome-wide heat maps or browsing on genome browser like IGV. So just getting through secondary analysis, you have a couple of options available to you that can really be reduced to 3 categories. One is that you do it yourself. The nice thing there is you have on-demand access and you control the pipeline and all of the parameters that go into it. The cons there is, as I said, we've been democratizing access to these technologies at the bench, which means bench scientists are going to need to learn command line coding. You're going to need to either build a high-performance computer for a couple tens of thousands of dollars or find access to a compute cluster. And you're also going to have to decide all the parameters, all the pipelines, all the packages that you want to put together. And then there's the question of reproducibility in terms of, was your data analyzed the same way as everyone else's? Can we compare head-to-head? To overcome some of those challenges, you could go with a collaboration and find a bioinformatician to work with, either another academic or working out of a core. This lowers the barrier to entry. You don't have to get your own equipment. However, the con there is that, as we've learned from our customers, many bioinformaticians have really long analysis queues. You can do your cut and run or tag assay in 4 days and sequence on the 5th day, but you may end up waiting 2 months to actually get your bioinformatician to analyze your data. They also may not have experience with cut and run and CUT&Tag, so you may have to teach them how to actually build your pipeline for you. And they may need training in your biological question if you're going to be using them to sort through the tertiary analysis that you want to do of gaining biological insight out of your data. Lastly, you can use a paid service. These are usually cloud-based programs. They work really fast, and some of them have pre-built solutions, but a lot of these have large annual subscriptions. We're talking about tens of thousands of dollars per year, and that may not mesh up with the number of cut and run or tag samples you're doing in a given year. Many of them are built for bioinformaticians. So even if you're using a cloud platform, you still may need to know how to code and assemble your own pipeline. And they're not specialized in epigenetics. The business model here is that you really have to be everything for everyone to garner the most number of users. And that's just not what we're trying to do here. We wanted to really focus on, the cut and run and cut and tag assays and deliver one type of solution for those particular people. And so in that spirit, EpiCypher, in trying to find a solution to our own bioinformatics bottleneck, really wanted to find a hybrid of those 3 options. Prior to this project, the way things looked at EpiCypher is, if we would get 60 cut and run reactions off of our NextSeq 2000, we had a handful of high performance computers in the lab. And if we wanted to analyze that experiments, you're looking at a roughly 24-hour turnaround time just to get the data aligned and back to you. However, those pipelines then spit out a variety of outputs where, if you want to actually understand how well your experiment did, you have a couple extra hours of work building reports and trying to assemble all the various different sequencing statistics into a single place so that you can make sense of how well your experiment did. However, on the cloud side of things, if you're engaging cloud resources, you can parallelize that high-performance computer, as Anand was talking about, and deploy resources at scale to have elastic computing available to you, so that, and then have it gather everything back up on the back end and deliver a report in a single workflow. And the nice thing there, as Anand was talking about, is that then you can align either 6 or 60 reactions, as we found in the deployment of our pipelines, that as long as you hit the recommended sequencing depth of 5 to 10 million reads, you're looking at results in as little as 90 minutes instead of more than a day. So with that desire in mind, we went on the search for a provider that could really help us build the solution that we wanted. On EpiCypher's side, we were bringing deep epigenetics expertise, rigorously optimized pipelines that have been used on over 30,000 reactions of cut and run and cut and tag to date, and a direct understanding of what our customers would want out of this type of analysis product. And We ended up finding DNAnexus. We really like them because they are a trusted bioinformatic provider, particularly in terms of the security of their platform and data storage, and also access to scalable cloud infrastructure that really allows us to deploy these pipelines and process the mountain of data that's starting to be generated. And with that, we developed CUTANA Cloud, which is a purpose-built platform for streamlined, secure, and reproducible cut-and-run and cut-and-tag analysis. So what does it actually do? Well, we talked to a lot of our customers to find out what the real pain points were in this bioinformatic process. And really, it all boiled down to secondary analysis. And people who pointed us towards secondary analysis really fell into one of two buckets. They're the wet lab biologists, and they're the bioinformaticians. And really, we heard universal support that this was the right place to target with the first version of the product. Largely, that's because for a wet lab biologist, there's a really steep learning curve to developing a pipeline, finding those resources, and getting your data analyzed. We've talked to people who have generated mountains of cut and run data and had yet to actually analyze any of it because they couldn't gain access to a bioinformatician. But interestingly, once you get to the top of that learning curve and you've figured out the secondary analysis on your own, bioinformaticians don't really want to do secondary analysis. It takes a long time. It doesn't really engage you intellectually, and they would much rather spend their resources doing the actual fun part of the data analysis, which is tertiary analysis, so that you can figure out what's actually going on in the data. And so to that point, we developed the workflow for what we wanted CUTANA Cloud to be. We really wanted it to be a streamlined, user-friendly experience that didn't require any code. We wanted the ability to upload data, importing directly from cloud resources like Basebase or AWS, so that you didn't have to download and re-upload gigabytes of files every time. Once you're on the platform, you annotate reactions with experimental metadata. We give you a FastQC report to make sure your FastQs are actually worthwhile of sequencing. And then you pick which reference genome we want to use, and you click Go. For version 1 of the product, we've got genomes for human, mouse, Drosophila, and yeast. Human has HG19, HG38, and T to T. There are select advanced parameters that you can fiddle with if you know what you're doing, but largely the whole workflow is turnkey to really remove the parameter paralysis that a lot of people experience trying to put together pipelines. We also collate a bespoke QC report to really tell you in one place automatically all the information you need to determine whether or not your alignment passed or failed, which is the #1 question our tech support people get, is just trying to figure out whether or not their experiment worked. And once you're satisfied with the quality of your alignment, you can then go and call peaks. You just have to choose which peak caller you want to use based on your expected peak size. And then we yet again deliver a peak calling QC report that's going to help you determine whether you chose the right peak caller for your particular data set. And after that, we have an on-platform IGV browser that helps you to determine pass or fail without having to download bigwigs on a failed experiment. And then you can download all your files in bulk and you're ready to go for tertiary analysis, which a lot of people have told us that yes, tertiary analysis is still going to require some coding, but it is a much more surmountable hurdle even for wet lab biologists to figure out the tertiary analyses they need, as long as they have BAMs, bigwigs, and bed files that they know they can trust. So with that, I just want to give you a brief look at what that process looks like on the platform. It is a project-based platform. Once the site goes live, anyone's going to be able to create an account, get on, and look at our gold standard data set so that you can get an idea of what the outputs of our pipelines look like. And then once you have an account with a billing account with us, then you'd be able to create your own projects, which are containers that can gate permissions for various groups. So if you have a collaboration, you can create a new project and set up a container that controls the permissions of what people are allowed to do with those data inside. As I said, we wanted to really diversify the ways people can get their data on the platform. So you can do local uploads, you can connect directly to your Basebase account, and we also have access to Amazon Web Services and from servers. Then kind of the key part of the new experiment workflow is to enter the metadata for all of your reactions. One of the problems that we personally encountered using cloud platforms in the past is that they really took a per experiment view of per reaction view of processing data. And we know from our experience at the bench that really the value comes in being able to look at your whole experiment in a single go. And so whether you're doing an 8 experiment pilot or 130 reactions that comes off of a P4 cartridge, you can put all those reactions into a single experiment, teach the pipeline what to do with each of those reactions, and then process them all simultaneously. On the back end, when your alignment is done, one of the things I'm really proud of is that we've built it so that you can get your run methods directly assembled. This is a Madlib style run method that we created for copy and pasting directly into your publication. And it's actually going to automatically change the outputs of this blurb here based on the parameters that you set up in your reaction details, and also if you fiddle with any of the advanced parameters in the run. You then want to make your way to the QC report, which is the place that's going to collate all the information you need to decide whether your run passed or failed. Particularly what we look for when we're trying to determine if an experiment was good or not, you want to look at your total reads, that you hit that 5 to 10 million sequencing depth, your duplicate read rate, seeing that it's less than 20%, and your unique alignment rate, that it's greater than 60%. And these are not dogmatic numbers. You're not going to fail if your unique alignment rate was 59%. But really, we want to see that if you're working with a data set, it's going to have more than 1 million uniquely aligned read pairs, and that nothing is standing out as a wild outlier. And really, as our director of genomics like to say, CUT&RUN and CUT&Tag are really multivariable assays. And so the passing or failing of a reaction, you really have to look holistically at the entire experiment to determine whether something worked or not. For example, here, if you're looking at E. coli reads, let me pull up this pen. If you're looking at these like E. coli reads right here, yes, IgG is outside the scope of what we would want here, but you really have to think of this in context of the fact of the output DNA that you're getting, knowing that a low abundance target like IgG is going to have a higher percent E. coli, and high abundance targets like H3K27 tri, that's going to have a lower than expected amount. And so you really have to think critically about what you're expecting to get out of this data before you dogmatically apply any of these criteria to your experiment. The other thing you're going to find in the QC report is automatic analysis of the SNAP CUTANA spike-in controls that EpiCypher makes. These are really useful for determining the success or the cross-reactivity of any post-translational modification antibodies that you might be using. For example, here, if you look at the left side here, you're looking at 3 different reactions of CUT&RUN that we performed, IgG, K4 tri, with 2 different antibodies. The one marked good here, when profiled against all the nucleosomes in the panel, only pulled down nucleosomes bearing the H3K4 tri mark, whereas a bad antibody that you might find if you just bought a commercial one randomly actually binds to H3K4 di, as well as some other trimethylation marks in an unacceptable amount of cross-reactivity that we would see. Lastly, the QC report outputs individually aligned transcription start site heat maps and gene body heat maps so that you can see whether you're getting the roughly expected localization of your targets to the expected genomic feature. H3K4 tri is a mark of promoters, and so we would expect that to show up around the transcription start site, whereas H3K27 tri is usually found more in intergenic and also silence genes. So if these were to be row-linked, you would expect those to be oppositional to each other. And then finally, when you're looking at your alignment, the best place to see whether or not things are working or not is to just pull up tracks in IGV and make sure you have the expected peak structure. So here we're looking at reactions of IgG, H3K4 tri, K27 tri, and CTCF. And what you really want to see is that the IgG is giving you a nice fuzzy line without any distinct peaks popping up. K4 tri is going to be showing up around promoters as it's a mark of active transcription. And then K27 tri is oppositional to K4 tri, showing you repressed genes and heterochromatin regions. So now that we're confident that our alignment went well and the data is looking as we would expect it to, then we can proceed with peak culling, which is a new analysis. I'm not going to walk us through that workflow, but you can largely expect the same experience that we got through the calling up the alignment. On the back end, when the peak calling is done, again, you're going to get a run method that you can directly copy and paste into a publication if you're using these data for a publication. And we're going to assemble a QC report that's going to read out the key features that you need to know from peak calling, namely the number of peaks that were called, the reads that appeared in those peaks, and the FRIP score, which is a measure of fraction of reads in peaks to tell you basically the signal to noise that you see in that data set. We also call up the top called peaks of each of the files, with those top peaks formatted such that you can copy and paste them directly into IGV so that you can quickly jump to the areas of the genome that you're seeing the highest signal from these marks. We built a peak annotation plot that is a really useful qualitative tool for assessing the replicability of duplicate reactions to make sure that the peaks called in those files are associating in the right proportions to the various gene regions found in the genome. And lastly, when you pull the peak calling files up in IGV, I'm really proud of the fact that we got it so that when you pull them up, the bigwigs are automatically associated with the bed files to give you the ability of really qualitatively interrogating whether or not the peak color that you chose is faithfully representing the data that you see in the bigwig. Because, as I said, just because you're getting more peaks doesn't necessarily that you did a good peak calling. So that is really the end-to-end value that CUTANA Cloud can offer right now. But that's just version 1 of the product, and we have lots of lines of development in mind down the road. For one, we want to make more sample compatibility, so we're going to be expanding the available reference genomes coming up, and hopefully get to the point where we can allow custom genomes to be uploaded for those of you working in more niche cell types. There's a view for the future to bring new assays onto the platform. I think DNA methylation and accessibility make a lot of sense, just as people really want to layer their multi-omic data and get a holistic view of what's going on with their cells. FastQ preprocessing is something that is going to make it into the next version. We really want, if you're using 2x150 sequencing, you're going to need to trim your files before bringing them on board. And eventually we'd like that to happen on the platform, as well as possibly including FastQ downsampling for those of you who may have over-sequenced your files. In the tertiary analysis space, we'll be working on row-linked heat maps, differential enrichment, Pearson and PCA. But really, the biggest driver is going to be customer-driven features. And that kind of falls into 2 buckets. The first one is compliance certifications. So that's where Anand was talking about security. We really want to get to the point where we can offer certifications to drive additional confidence in the platform. But those are going to be driven on a need base as particular customers request those. And also just any feedback that people want to submit on the platform to help steer our development efforts. We really want this to be a user-driven development process so that we're delivering the things that the people who are using the platform want it to be. And on the note of compliance certifications in particular, in order to get the product to market for the most number of people as quickly as possible, right now it is going to be US only because we have not gone through the process of ensuring GDPR compliance. And it's also going to be research use only because we would be interested in pursuing HIPAA certification in the future. So with that, the real take-home messages are epigenomics is essential to understanding the cellular identity and gene regulation. Bioinformatic analysis is an increasing bottleneck for cut-and-run and cut-and-tag due to the expanding throughput of wet lab scientists. And researchers have told us that they really need an intuitive experience in a secure environment with fast turnaround. And that is why we have built CUTANA Cloud to help people accelerate their epigenomic discovery. So with that, I'd just like to thank everyone at DNAnexus who's touched this project. It's been a long haul for us. Amy Leung was the first one to really deploy our pipelines and get us excited for what DNAnexus could do with our pipelines. But Allison Rieger really brought it home and did just tons of work helping us to build a really robust and valuable pipeline that puts things together in an intuitive way for non-bioinformatic users. Cecile and Teresa have been good shepherds for the program. Mark, Alice, Sam, and Roger have really done a good job managing the program. Sudhir has been an amazing help for development, along with David and Vishal, to really build the vision of what we wanted to be here. And Sean and Jeff, there was a lot of negotiating involved that went through this process, and really everyone at DNAnexus has been immensely helpful getting us over the finish line. On the EpiCypher side, for product help, Andrea Johnstone and Ricky really to help steer this process in a knowledgeable way. Of course, all credit goes to Ali and Brian for the building of the core pipelines, then really teaching me how to be a bioinformatician as well. Nathan for running everything, our customer service team for learning how to be SaaS operators, and everyone in R&D has touched this thing. We have really beat up the platform to make sure that it passed the standard for what our own employees would want to use. And now we're excited to get to the point of let other people using it. And finally, none of this would have been possible without the generous support that EpiCypher has received from the NIH. The SBIR program is truly a great lever for driving innovation in the private field. And we're truly grateful for the support that we've gotten. And
we'll now begin the Q&A portion of the webinar. Our first question, do you think that cut-and-run and cut-and-tag will eventually phase out ChIP-seq?
I'm a little biased, but I have not found applications yet where cut-and-run and cut-and-tag don't perform in a superior fashion to ChIP-seq. Is it going to fully phase out? No, I think a lot of people, scientists don't really like change. And what we found is that if you've got a system set up that's working for you, there isn't a lot of impetus to change. I do think there's a lot of value for people to consider changing purely because cut-and-run and cut-and-tag dramatically reduce the cost per reaction and the throughput that a single scientist can do at the bench. So the value isn't just in the data quality; it's also in the quantity of data and throughput that you're going to get out of your researchers at the bench. So I think, yeah, eventually everyone's going to come around and see the light.
Thank you. One moment, please, while we gather questions. Our next question, what kind of normalization controls do you recommend for cut-and-run or cut-and-tag samples for downstream analysis?
Yeah, that's a great question. So we've tried to provide tools to allow people. So that's a big question. And I don't think it's going to be the same answer for everyone with every experiment. It's certainly not going to be. We've tried to provide tools to assist people in that. We have the E. coli spike-in our cut-and-run workflow. We have our SNAP CUTANA nucleosomes that are useful in both cut-and-run and cut-and-tag. And those tools are out there to let people experiment with them. But largely, I think what we found internally is that standard RPKM normalization, even when you're doing, things that are going to affect a marked genome-wide, you can largely see dose-dependent effects just with that standard normalization. I would defer to our tech support team for a more nuanced answer beyond that. But really, our view is that, we don't want to pick the winner in this regard. We just want to provide the tools that will allow people to mess around and figure out what's going to be best for them.
Thank you. Our next question, do you have internal controls available for TF, which I believe is transcription factor analysis?
Yeah. So we are capable of making controls similar to the SNAP spike-ins that we use for our lysine methylation panel. The problem with transcription factors is that there's a lot of them, and trying to drill down to which ones are going to be popular, it's not a really commercially viable path to take. I think the best path forward, if you did want an internal control for transcription factor, would be we do sell FLAG and I'm blanking on the other one, but we sell epitope tag antibodies that come with spike-in controls. So if you were able to recombinantly express your transcription factor with an epitope tag, you would be able to use our spike-in controls to determine the specificity in that regard and use those for normalization.
Thank you. Our next question, for testing whether an antibody is good or bad, how do you create the heat map that you showed?
Oh, the genome-wide heat map? So the transcription start site heat map, those heat maps are automatically generated by the pipeline using the bigwig files that the pipeline generates. I think for a more useful visualization of that kind of data, you're going to want to download those bigwigs, and internally we use deep tools. And this is kind of the point I was getting to earlier where we identified secondary analysis as the key place that we wanted to get started with this platform, because as we found internally, we have 3 bioinformaticians at EpiCypher, and once the onus of doing secondary analysis was removed from them by having access to these pipelines, most of the wet lab biologists in our lab with an hour's worth of training, were able to deploy deep tools on their own laptops and do these types of tertiary analyses, with minimal support from a trained bioinformatician. So yeah, to the question of like making those heat maps, you're going to want to download the bigwigs and deploy deep tools, but that can be done on a laptop instead of needing, a high performance computer.
Thank you. One moment, please. So our next question, how would you compare your pipeline to NF Core?
I am not an expert in NF Core. I would say, so our pipeline, I'm not sure if this is going to be a good answer, but our pipeline is built off of Bowtie 2, is like the core part of our analysis pipeline. Yeah, I'm going to have to follow up with that one on email because I'm not familiar with NF Core.
Okay, thank you. Our next question, how does CUTANA Cloud perform the normalization to IgG since we don't use input anymore?
So IgG is useful sort of qualitatively at the alignment stage to just make sure you're not getting really strong peaks. But the actual computational use of IgG comes in the peak calling, because most of the peak callers can use the IgG control BAM file as the definition of background. So if you have really high peaks in your IgG control that match the amplitude of the peaks that you're seeing in your experimental sample, the peak caller will not call a peak there. So that's how the IgG is used. It's not really normalization, but that's I think the spirit of the question there is.
Thank you. Our next question, is single cell cut and run or cut and tag possible?
Single cell cut and tag is definitely possible. And the reason that cut and tag is necessary is because really you want that tagmentation step to happen in a single step. EpiCypher has been largely focused on technology development of the core technologies, and so we've kept our focus on bulk assays. But there are other providers out there who are developing single cell cut and tag offerings that we've been supporting their efforts to do so.
Thank you. Our next question, for antibodies that don't have commercial cut and run options, would you recommend adding spike-in controls in those conditions?
There's two parts there. So for a post-translational modification, particularly if it's a lysine methylation antibody, I definitely recommend using our K-Metstat panel, the SNAP CUTANA K-Metstat panel, because that'll help you determine the specificity of that antibody to related lysine methylation states. If you're working with other histone PTMs, I think we're roughly targeting next year to launch a new panel that's going to cover lysine acylation states. So you can keep your eyes out for that. But beyond that, if you're working with like transcription factors or post-translational modifications that aren't related to these panels, your best bet is to just buy what we typically do when we're doing R&D or providing services is we'll buy 3 to 5 antibodies to a particular target to, different vendors, different clones, and then just compare what the sequencing results look like and try and make sure that the tracks that the antibodies are producing match the expected biology of the target.
Thank you. Our next question, which yeasts can be analyzed right now?
The genome that we have for yeast right now is Saccharine, which I'm ashamed to say I don't know which strain of yeast that is, but that sort of falls into the category of a very near-term development plan, is to build a way for people to bring custom genomes onto the platform so that we don't have to pick the winners. And if we want to have validated genomes on the platform to give people peace of mind that everything's going to work. But we also know that people are studying all kinds of organisms and doing epigenomics in those organisms. And so the plan for the future is to be able to upload your own reference genome if we don't have one available for you.
Thank you. Our next question, for the tertiary part of the analysis, what approach does this platform use for getting the difference between two data sets, and how reproducible is that approach?
So right now, really the only tertiary feature available on the platform is the fact that we have the integrated IGV browser. And that's really just for the sake of being able to look at your tracks and see that you have enrichment over background and that peak shapes are, what you would expect. It's the web version of IGV, so it even lacks some features that I would really want for doing figure generation, like automatic group scaling. So I really use the on-platform IGV for the sort of qualitative assessment of, did my experiment pass? Are these files worthy of tertiary analysis? And then at the current moment, we then download everything and do tertiary analysis locally. So for the question of like doing differential enrichment, we don't have support for that now. I'm not even yet versed. We haven't really picked what we want to support in terms of which packages, because there's a number of different options for doing differential enrichment. But in terms of asking the question of how is one data set different than another, that would probably be a good first step, is doing differential enrichment.
Thank you. Our next question: what peak callers do you recommend, and do you have suggestions for ideal peak calling controls?
Yeah, so the platform currently supports max2 narrow and CISR2 narrow and broad, but really, I think functionally, in terms of the thousands of reactions that Cypher has done, we really use max2 narrow for just about everything except for really broad peaks that you would expect to be broad, like histone 3 lysine 27 trimethyl or H3K9 di-and-tri. Really, max2 narrow is kind of a workhorse for us, and we've found that it generally gives the best fidelity to the data when we're looking at the peaks that are called against the structure of the bigwig.
Thank you. Unfortunately, that is all the time we have for today. We'd like to thank Keith Mayer of EpiCypher, Rao of DNAnexus, and our sponsor, DNAnexus.